Hi friends, this is Miss Medico channel, and today we are going to start a video lecture about types of transduction. So far we had already discussed about the transduction introduction, and we already know that what is bacteriophage and how it's able to carry the bacterial DNA from one bacteria to another bacteria. So this already we had discussed. Today's video lecture is about types of transduction. So let us start. We have got two ways by which bacteriophage is able to carry this genetic material from one bacterial cell to another bacterial cell. So there is generalized transduction and there is specialized transduction. Okay. So, firstly, we will discuss the generalized transduction. We already know that this is bacteriophage. This is bacteriophage. Our main point of discussion here is bacteriophage. When it infects the bacterial cell, what happens? What happens? First of all, adsorption occurs. Then there occurs penetration. First of all, adsorption, and then penetration of this phage into the host's cytoplasm occurs. This is bacterial cell. This is bacterial DNA. We make it circular and it is circular. And this is the phage DNA. After adsorption, phage DNA comes into cytoplasm of bacterial cell. What happens next? This phage DNA is transcribed. I will make it again bacterial cell, and this phage DNA uses the RNA polymerase of the bacteria, and this is transcribed, makes its own copies. This is phage DNA in the host's cytoplasm. This is transcribed, replicated, makes its own copies, and translated later on into CAPSID. This is then this transcribed. This phage DNA is translated, replicated into this host cytoplasm. What happens to the bacterial DNA? At the same time, this bacterial DNA, in presence of viral DNA, this bacterial DNA undergoes lysis, means it is destroyed. Bacterial DNA is destroyed and repressed in presence of phage DNA. Repressed. Okay. And the bacterial cell undergoes lysis, and this phage DNA is then come out into capsids. This happens in generalized transduction. But sometimes what happens, pieces of bacterial DNA are left intact. Pieces of bacterial DNA are left intact if these pieces are equal to phage DNA. Then but sometimes what happens is that the phage DNA in the host cytoplasm is transcribed, replicated. It makes its own copies. It uses the RNA polymerase and translate it into capsids. What happens to the bacterial DNA? It is destroyed and repressed. But sometimes what happens is that the pieces of bacterial DNA are left intact. If these pieces are equal to phage DNA, then accidentally, if they are equal to phage DNA, then accidentally they are packed. This bacterial DNA is packed into capsid. Accidentally, sometimes it happens. Normally just phage DNA gets released and this bacterial DNA gets repressed. But sometimes what happens, these bacterial DNA are left intact, and if these pieces are equal to the phage DNA, they are accidentally, I'll make it more prominent, accidentally what happens, they are packed into capsid. So I will make capsid here, and accidentally this capsid doesn't contain this phage DNA, but this contains this, what we call the bacterial DNA. It happens in generalized transduction. Following lysis from the bacterial cell, what happens is that one phage will come out with bacterial DNA. So when this bacterial DNA in the capsid, when it infects the another bacterial cell, what happens? This is bacterial cell, and this bacteriophage, actually we call bacteriophage, with the bacterial DNA will get transported into the another bacterial cell. That is how the bacteriophage with the DNA of the bacteria is transported to another bacterial cell. And if there is analogy between this bacteria and this bacteria of the recipient bacteria, if there is some homology between these two, what happens is that gene on that bacterial DNA will form a protein. Obviously जीन का काम ही है प्रोटीन फॉर्म करना, that the recipient will not have. So actually such a protein may be that protein that will inactivate an antibiotic. That is how the bacterial DNA is transferred through bacteriophage from one bacterial cell to another bacterial cell. यहाँ पे जो phages निकलेंगे जब लिसिस होगी इस बैक्टीरियल सेल की, यहाँ पे और भी phages निकलेंगे, लेकिन जिनमें वायरल डीएनए होगा। ये जो मैंने सिंगल लाइन से बनाया यही डीएनए होगा, लेकिन एक फेज निकलेगा जिसमें बैक्टीरियल डीएनए होगा, और वो जब इन्फेक्ट करेगा अदर बैक्टीरियल सेल को तो वो इस तरह से अपना जो मटेरियल है, जेनेटिक मटेरियल, ट्रांसफ़र करेगा टू रेसिपिएंट बैक्टीरियल सेल. दिस हैपन्स इन generalized transduction. So what happens? Number 1, adsorption occurs, adsorption of bacteriophage. Number 2, penetration. Here viral DNA is marked as a thin line and bacterial DNA as circular ring. You all know now. Then happens is that destruction. Number 3 occur is destruction of what? Bacterial DNA. Number 4, phage DNA undergoes replication. Phage DNA undergoes replication. Number 5, what happens is that if some pieces remain intact, some bacterial DNA pieces remain intact, they are then transferred to capsid and after lysis this comes with phage comes with this bacterial DNA and it will infect another bacterial cell. This is all about the generalized transduction. Next video lecture will be specialized transduction. Hope you have understood this video. If you have any query, please comment in the comment section.